Yeah, yeah. I'm just getting it going. Just going to the wrong page. All right, so as Mark mentioned, I'm going to be describing our software package called BrainSuite today. And what my objectives are to basically introduce you to the suite of tools and what it does, and to provide you with an overview of the major components of the software. And we'll do hands-on training after I've described in the presentation, so you can use the sample data sets we provided. And then if you want to try out some of your own data, we'll stick around for most of the afternoon. And I've also, my research assistant is here as well, and we have another person from our group that'll be joining for the labs to help out to show you how to use the software. So if you're not familiar with BrainSuite, it is essentially a collection of image analysis tools that we've been developing for quite some time. And it's originally designed for structural imaging. We've added tools for doing diffusion MRI processing as well. And it essentially, there are about 4 main components that I'll talk about today. The first is a method for automatically extracting the cortical surface from a T1 MRI. The second part is to perform a combined surface and volume registration. And then we also have the tools for doing the diffusion processing data, and then also a lot of tools for exploring and visualizing the data, as well as editing and performing delineations. And the software does run on Windows, Mac, and Linux. We don't have the Linux GUI built for release yet, but it can be built, and we'll be distributing that later this year. I don't know how many of you use Linux by preference, but okay. Well, if you're gung-ho, you can compile it yourself as well, but we're hoping to get a distribution out soon. So this project started when I was a PhD student, and the first release was basically when I finished my PhD, and I did that work with Richard Leahy, who I've continued to collaborate with ever since. And we've continued to grow the software, and I want to acknowledge that this is the work of a great number of people who we've worked with and continue to work with. And so I've listed a few of them here. In particular, Anand and Chitresh have been, Anand Joshi and Chitresh Bishan have been, actually I should add, he just finished his PhD, I forgot to update that, have been really essential in developing some of the core methodologies, and as has Justin Halder, who's a professor at USC as well. And then Shantanu, who's here, has been developing our statistical tools that I'll talk about a little bit. So I wanted to describe some of the highlights about BrainSuite, and this is sort of to get at the question of why you might want to use it, because there are obviously a number of other terrific tools out, and you may have your own preferences for what you want to do. But the main benefits of our tools that we see and that our users often report to us is that it has a lot of flexibility for performing interactive processing. It has a lot of powerful visualization capabilities. We have some novel algorithms, including a method for performing cortically constrained volumetric registration, which tries to combine the best of surface registration with volume registration. We also have a custom atlas that was developed by Hannah Damasio in collaboration with us. And you have the ability in the software as well to develop your own atlases if you want to look at a particular age group, for example, or you might want to look at a different species even. So we have examples of monkey atlases and cat atlases that have been developed. We also have some unique methods for performing diffusion modeling, as well as correcting for distortion in diffusion data. And we have some novel tools for exploring connectivity visually. And I'll also add that we've gone to a lot of effort to ensure that the software that we've written, which does work on Windows, Mac, and Linux, provides consistent results across the platforms to within levels of machine tolerances, which I think is really important, obviously, if you want to try and work in a mixed platform environment. And then finally, we are open source as well, which I think is important in terms of the important movement of trying to have open science and having open methods, so you can actually look at the code if you want and see exactly what's going on, as well as develop from it yourself if you would like to do that. So the motivation behind what we do, and I think this is fairly review for everyone who's at the advanced training program, but we're interested often in looking at comparisons across different brains or different points in time. And so to do this, it's really critical that we have good spatial correspondence across the data. And then once we do that, we can look at different aspects of the sizes of structures or the thickness of the cortex, or complexity, or various other measures of things such as the white matter structure, and then look at how these change over time, or when there's some disease present, or some other aspect of the individual or group that you want to study. So our goals in trying to address these types of problems is that we want to try and automate things as much as possible, but we also recognize that automated methods are imperfect, and so we've tried to provide the facilities to make corrections and intervene when necessary in the processing stream. And we've also tried to provide the tools that allow you to perform and streamline the tasks of analysis. And this is an ongoing work, and so we are continuing always to try to improve the software. And so if you do use it, we appreciate hearing feedback from users about what they might like added or what doesn't work for them. So as I mentioned, a lot of what we do really relies upon image registration, and there are essentially two schools of image registration that are commonly used. One is the volumetric approach, and the other is the surface-based approach. And there are a few methods that perform both. Ours, I think, is fairly novel. And so we're really interested, first of all, in the surface models because you get better representations of cortex. One issue you have with volumetric registration is that you don't always align the sulcal anatomy well and the cortical anatomy well because there's so much variation in the cortical areas. And with surface-based models, you're actually representing the fine folds of the cortex with a surface, and then you can match the folds between subjects or between different time points or to the atlas. And so as you'll see later on, the advantage of our approach is that we do get the good matching of the cortex from the surface methods, and then we can also match the subcortical areas with a volumetric approach. And so essentially this is one aspect of the kind of workflow that we do in the software. Use my mouse, right? So we can follow essentially two streams. One performs the surface extraction and the registration and automated labeling, and the other one performs the processing of the diffusion data, so that we can extract things such as tensors and ODFs and perform tractography. And then we can combine these together to look at connectivity and look at different aspects of the structure of the brain. So I'll start off first by describing the cortical surface extraction component, which is the oldest portion of the software. And it essentially has about 10 different steps. I've summarized them here. And it can run on a typical size data set. You can probably process it in under 10 minutes to extract a model of the inner and outer boundary of the cortex. And so in the software, we have this integrated in a GUI where you can set different parameters for each step of the processing. And we'll run through an example of extracting that during the lab today. And I'll go through a little bit about what the different steps are, just so you understand what they're doing and why they're important. You may be familiar with some of these aspects from other types of software that you've used. One of the big problems, obviously, that we always face in brain imaging is removing the non-brain tissue. So we have an algorithm that works rather quickly to identify the boundary using edge detection, and then it cleans it up using mathematical morphology operators. And this only takes a couple seconds to run. So it's very fast, and you can adjust the parameters. The parameters can be a bit tricky to tune, and we are working on methods for automating that parameter selection, which we're hoping to have sometime, well, I won't make any promises, but sometime soon. We had some encouraging results recently, so I just got an email about it this morning, but it may take some time before it makes it into the usage. The second aspect, once we have the brain extracted, we can actually extend that back out to identify the skull and scalp. And so this is useful in certain types of problems, for example, in EMG or EEG source localization. There are some groups that are using this to do head modeling for TDCS or other types of neurostimulation, where you want to simulate the electric fields in the brain. We don't use this a lot for the work that we do, but if it's of interest to you, it is built into the software. And then, so once we've extracted everything, extracted just the brain out of the head, the problem that we often run into, as you've surely encountered, is that images often present a great deal of non-uniformity, particularly depending on how they're collected. And so we have a method performing bias correction that essentially looks at histograms throughout the image, and it compares these histograms to the global histogram to determine how much the image intensity has shifted locally. And so it then performs a correction step where it estimates a correction field based on a B-spline fitted to those data, and then that can then be applied to the image to normalize the intensity. And so once that's done, we can then proceed to do a tissue classification step. And here what we're doing is doing an analysis of the histogram as well as the local neighborhood of each pixel to identify the different tissue types according to gray matter, white matter, and CSF, as well as partial volume voxels, which lie at or essentially the voxel is too large to capture the fine boundary of the tissue, and so you have a combination of tissues in those areas. And so we estimate at each voxel what fraction of tissue is there, and this becomes important later when we're doing boundary detection as well as computing cortical thickness. So we'll reuse these outputs later on in the software. So once we've identified the white matter and the gray matter, the next thing that we want to do, sorry, that was just showing the difference between the maximum likelihood and the maximum posteriori segmentation, which ensures basically that we have contiguous regions of tissue. So once we've identified the white matter in the brain and the gray matter in the brain, what we're trying to do next is find the boundary between, we want to just extract the cerebral cortex. So to do that, we need to first identify the cerebrum. And so we use AIR, which was developed by Roger Woods here at UCLA, and we have that integrated into the software. And we fit a fairly rough atlas to the data. So we're not doing a really detailed segmentation here. We're mainly just finding the cerebrum and the cerebellum and also labeling some of the subcortical areas so that we can ensure that they get kept in the image that we extract. And then we combine this with a tissue map to get out a boundary of the white matter, so basically the inner boundary of the cortex. And so this gives us an initial surface, but the problem if we just generate a surface from this is that the classification's imperfect, the data resolution may not also capture the shape well, and so we wind up with a problem with topology errors. So essentially we can get small handles or holes going through the surface, and we want to model the surface as being essentially the equivalent of a sphere, or you can think of it as a two-dimensional sheet wrapped around the cerebrum. And so we have an algorithm based on graph theory that identifies all these topological handles and holes and performs a minimal set of corrections to remove them. And so you can see some examples here where there was a small defect that we corrected. And so what this does is ensure that we can describe the surface of the brain using two-dimensional coordinates because it is topologically equivalent to a sphere. And this becomes important when we're doing mapping because it means we can have a consistent coordinate system between one brain and another, and we just need to compute the transformation from one surface to the other. And then some of the other steps essentially perform some filtering to make sure that the object that we generate is smooth. And these are, I've shown the representations here as surfaces, but in fact they're all happening in the voxel space. So they occur very quickly, only take a few seconds to run, and ensure that we get a nice smooth intercortical representation. And so once we fix those, we again, we generate the surface after these corrections, and it has the topology that we want. And then we have a process that essentially expands this out to find the peel boundary by looking at where the tissue classification results show the boundary between gray matter and CSF. So essentially each vertex pushes outward while trying to stay smooth. And so this also, if we look at a cross section, you can see how it follows the cortical ribbon. And it also gives us an estimate, a crude estimate of what the thickness is, because each vertex does move some distance to get out to the boundary of the cortex. And so we can use that distance, which we describe later as a linked distance, as an estimate of cortical thickness. But it actually turns out it's not a very good one. So we have a much more improved method for finding this, and I'll share some results about how much better the new method is compared to using this type of approach. So the final thing that we do is that we split the hemispheres in half, which is fairly easy to do for us because we already had the step where we registered our crude atlas to the cerebrum, and that had left and right separated, and so we apply that to the mesh, the surface mesh that we've generated, and we do this for the inner and the outer ones. And so now we can look at the left and right hemispheres separately, and we'll apply these in the registration step. One other thing I just wanted to mention, this isn't in your slides, but if you do want to print a 3D model of your brain or someone's brain, you can actually export fairly easily to an STL file and 3D print it. So this is a model of my brain that someone printed for me a couple of years ago. Well, she didn't print it for me, but she printed my brain a couple of years ago. But it's actually kind of small. So I'll just point out this isn't life size. But
can you print it with detachable hemispheres?
I think, yeah, you could do that. Yeah, yeah. A friend of mine just printed one for another friend's 30th birthday. So, and I've seen them. You can do much larger. And I do actually have some clinical collaborators who've used this to print some of their cases. So I had one collaborator who emailed me and asked. He thought it would be like impossible, and I pointed out, or like it was a strange thing to request, but it was already built into the software. So he was printing a brain within about 15 minutes of emailing me. So it is some rather fun aspect of the software. But moving back to the more serious components. So we have, once we've generated the left and right hemispheres, we can actually use these to perform the surface and volume registration process that I mentioned. And so this is the series of steps. This is a method that was developed by Anand Joshi, and he's continued to refine it over the past several years to make it more reliable and to work with an atlas that we have that was labeled by Hannah Damasio at USC. And so the process, well, it relies heavily on having a good, accurate atlas. And so we have two atlases that are built into the software. And the first is based on the well-known Colin brain. And it's been labeled into 98 different ROIs. You could also reparcellate it yourself if you wanted to create your own ROIs. We have a number of sulcal landmarks on each hemisphere, 26. And but there's some aspects of the brain that may not be as representative as the general population. So when we were developing the software, Hannah Damasio wanted to use a different brain, and so she selected a brain that she felt was more normative, more representative of, and would provide a better registration target for the methods that we were using. And so she's labeled this one in greater detail. The scan is also from a high resolution 3T scan. We also have diffusion data for the same subject that was acquired at the same time, which we haven't built into the software yet, but we will be using it in the future for some of the new methods that we're developing. Just a second. So those are the two atlases, and as I mentioned, you can customize them or you can create your own. We have tools for building your own atlases that are available separately from our website. So the method that we use for doing the registration is based on flat mapping, which is a technique that's been fairly widely used in brain imaging. And essentially what we do, so we have the two hemispheres already split. We have this boundary essentially at the corpus callosum or the inner hemispheric fissure. And we map that boundary to the boundary of the unit square. And this changes our registration problem from being a 3D problem to a 2D problem. And what you see in the image is that we've taken the curvature of the surface and put it into the unit square. So you can kind of make out the temporal lobe and the frontal lobe if you look carefully. So that's, I believe, the frontal lobe, and this should be the temporal lobe down here. I can't quite see it at this angle, so hopefully I'm pointing at the right spots. So what we do in our approach then is that we can match the curvature maps from two different subjects in the square space. And to do this in a reliable way, what we first do is we smooth the image to different resolutions. So at the most smooth resolution, we're matching the major folds of the cortex, which are more consistent across subjects. And then once we've established the correspondence for those, we refine it progressively through different levels until we're finally refining at the original level of the cortex. And so what this gives us then is a map from our subject to our atlas or vice versa, so we can take the labels. This is after we do a deformation. We can take the labels from the subject and then project them through these spaces to get the labels on the cortex. And this does a reasonably good job, but we have some additional improvements that we apply, which basically try to push the labels on each gyrus down into the sulcal valleys. And the reason for this is that the initial labeling when you do the label transfer is imperfect. You can kind of see in the folds here where the boundaries aren't following the curvature of the sulcus. And so we have a method that uses a geodesic flow to push them into the boundaries, and so they line up much more nicely with the curvature and the geometry of the individual subject that you're looking at. So that gives us essentially our final process for all the surface registration. And so we have, if things have gone well, we have a good map of one subject surface to another subject surface, or from the atlas to the subject. And so what we want to do then is we want to also label the volumes, because obviously the surface maps don't tell you anything about what's going on inside of the cerebrum. And so what we do for this is that we actually take these flat maps of the hemispheres and we map them onto the unit sphere. And this is a fairly straightforward mathematical mapping to get from this space to the next. And the more interesting part here is that we can also use a method called harmonic extension that maps everything that was inside of the brain hemisphere to be inside of the unit ball hemisphere. And so now what we've done is we've taken things from a non-matching 3D space to now where both brains are represented, both the cerebra are represented as spheres or balls. And so this gives us a mathematical space in which to perform an alignment so that we can find a one-to-one correspondence between every point in this sphere to every point in this sphere that matches the intensity profiles of the two images. And what's important about the way that we do this is that we can constrain the surface so that the surface points don't move. So essentially we keep the registration of our surface, and this is maintained in the surface space, the volume space, and the spherical space, but then we're allowed to move the points interior to that sphere or interior to the volume so that they line up. And so it gives us a way of getting a consistent mapping between the surfaces as well as the volumes. And so this is an example of where we've done the volume label transfer. And so we have the atlas with the surface labels as well as the volume labels, and they've been mapped nicely onto the subject. And so that's essentially how the labeling component of the software works. And then another component that's built into this software component of the software is also an improved method for measuring cortical thickness. And rather than relying on the link distance measure that I mentioned earlier, it computes thickness measure based on an anisotropic Laplace equation. And this improves on some of the other approaches that have been used for the Laplace approach, which is essentially you're modeling the flow of heat from one surface to the other, and the shortest path between them gives you the thickness. And what we do in our approach is that we've modified that to also incorporate the measure of partial volumes in the object so that we know what fraction of gray matter is in the tissue. And so I don't have the math in the slides here, but what we see when we look at this comparison, so this is an example of comparing the cortical thickness maps that were computed by registering 186 subjects with our method and mapping 3 different types of thickness measurements and comparing it to a classical map by von Economo. And so you can see with the link distance measure, it really doesn't correspond very well at all. But when we look at the more traditional Laplace equation, isotropic heat equation, it maps pretty well. And when we look at the anisotropic Laplace equation, it maps really well. There's really great correspondence, I think, between what we get here and what the classical map is. So if you are using our software for cortical thickness measures and comparisons, then I would really recommend running this process. It takes a while to run, but it's worth it for doing these types of studies. So I'll just mention this stage of the software produces several outputs. We have, as I mentioned, the labeled surfaces and labeled volumes. It also will map. We have sulci that have been labeled in the atlases, and those will automatically get mapped to your subject data as well if you want to perform measures on those. We also have tools if you want to interactively delineate the sulci yourself, and then you can use those as constraints in the registration process if you want to improve the registration, or if there's some particular area that doesn't register well, or if you have a subject with some abnormalities that you need to constrain better. And as I mentioned, we also get the cortical thickness maps. We also have these measures, or these maps, in volumetric space from the subject to the atlas, as well as the inverse maps. And so if you have data in one space and you want to map it to the atlas, for example, diffusion data, which we do a lot of mapping of those data sets, you can use these to transport them into the atlas space and perform studies there. And then we also have the Jacobian maps, which essentially tell you how much deformation it took to get from one voxel in the atlas to your subject. So essentially how much change of shape there is locally. And so by taking the determinant of those measures, we can do studies on relative sizes and relative shape change. And so this is how we do tensor-based morphometry with our tools. And then finally, we also generate tables of outputs in spreadsheet form that you can use to look at things such as the gray matter within each ROI, or the total volume of the ROIs, or the surface area of a particular patch on the brain. And we have, as I mentioned earlier, we do have a statistical toolbox that's written in a combination of R and Python that provides a lot of the facilities for doing group analysis. This is an ongoing development, and at the moment we have a number of statistical methods that are in there. And so you can define a table with all the metadata for your subjects, or your other measures from other, whatever other attributes about the subjects you want to compare, and put these into the tool and define your models and do comparisons. And so we have details. It's on the website if you want to learn more about that. And this is all open source as well, and it's extensible because it's R and Python, so you can develop your own statistical tests in it. And then finally, these in the registration, I'm going to show you some results. These slides aren't in your talk. These are preliminary results, and they're not yet quantitative. So I didn't want to put them out, and so take them with some caution that we haven't completed the study on this. But so these are just, this is an initial comparison we did to try and see how well we were doing at registering the data. So we took 843 subjects from the human connectome data set, and we registered those data with both FNERT and our SVReg tools. And then to look at the correspondence, we thought it would be interesting to look at the FA maps that were in each subject and see how well those aligned. And so we remapped the FA using the transformations computed by both software packages, and then we averaged those across the subjects. And so what we see, so in this region of the brain, hopefully this is visible. Find my mouse. So you can see that we didn't do as well in the cerebellum, which is outside of our surface boundary, but in the areas where we're interior to the cortex, you can see we have much crisper boundaries in the white matter. So it suggests that we're getting better alignment of the white matter. And again, this is only using the registration of the T1 data. So we're not using the diffusion data in the registration process. If we did, there are probably ways of improving both alignments. If we look in higher up in the brain, you can see hopefully this is visible on the screen. But here in the cortical areas, we also get much better, much crisper boundaries. So I think this is suggestive that the results that we're getting from the registration are an improvement over the methods that use volumetric processing alone. And so this is something that we're going to be exploring in the next few months to do some validation on our methods. So that actually leads kind of nicely into our diffusion pipeline, so which is essentially designed to get the diffusion data into the same space as the T1 and allow us to compute a variety of diffusion models, as well as perform tractography and do some initial connectivity analysis. And so what we do in our registration process is, again, we're trying to find good anatomical correspondence. And so we want to map the T1 and the diffusion data. And the problem that we run into, depending on how you collect your data, is that you can have really bad distortion often in DTI data due to certain aspects of the EPI sequences that are used. And these can be corrected in some cases with field maps, so you can use some special acquisition techniques that also do a better job of acquiring the data. But you don't always get data that is what you want. So the method that we developed relies on the T1 as essentially providing a reference of what the anatomy should look like. And it performs a registration that resamples or corrects the EPI sequences by registering them to the T1, but it does it in a way where it's constrained to the direction where the distortion is greatest, which is in the phase encode direction. And then we resample all the data into the space of the T1. So you can see, in this image, areas where it does well. It can't correct every problem. If you have really bad pileup, it's going to stretch that out. So not all signal is recoverable, but it does a decent job and performs similar to field maps. The software that we have also will apply field map correction if you have field maps. And so this was an example of the correspondence between the diffusion data and the T1 data. And you can see as well the ODFs, which are the orientation distribution functions. I think you all had lectures already on diffusion, but if you had questions on what these are representing, I'm happy to describe it. We'll go through in the lab. We'll take a closer look at the ODFs and the tensors. So I'll just point out in our software we do compute a number of parameters. We compute fractional anisotropy, mean diffusivity, color FA, and other measures. We also compute ODFs using the FRT approach, which is a traditional approach, as well as the FRACT, which is a method that Justin Halder and Richard Leahy, who are our collaborators on the project, developed, which provides improved accuracy and better angular resolution compared to the FRT. And we've also recently added a new method, which was just released yesterday, as you all know, which it incorporates as well 3D Shore, which is a reconstruction method that allows for more flexible sampling schemes. And so it can accommodate things such as multi-shell data, whereas for these methods, you have to separate out the B shell that you want. So we have some tools in the software for visualizing the diffusion data, and we'll take a look at those in more detail later this afternoon. And we also have tools for doing fiber tracking, and so I think you all saw a lot about tractography already. If you hadn't already seen it, you saw a lot yesterday and today. And so in our current implementation, we have deterministic streamlined tractography. We are working on doing probabilistic as well. And essentially it works by placing a seed point at various places throughout the image and then following the most likely path through the data. And so if we seed a lot of points, we can do some pretty impressive visualization of the tractography. This is just an example of how the tracks grow from the seed points. And this is another example where you can see it growing within the cortex. So you can see how we've mapped the diffusion data into the same space as our T1 data to have everything co-localized. And then we also have tools for exploring connectivity by essentially taking the tracks that we generated from the tractography and looking at which labels in the atlas they connect to. And I want to mention that what we have currently implemented is a fairly simple, for the measures, it's a simple count of how many fibers go between areas, which isn't a good way of looking at structural integrity, but we're working on some methods that will perform more robust measures of those. But it's still a useful tool for exploring the data and for looking at the pathways that are identified in the tractography. And so we'll look at that in the lab as well. All right, so that really captures what I wanted to say about the software so far in terms of the methodology. I did want to talk a little bit about what we're doing with it and how we're applying it and how we're trying to make it easier for people to use the software in larger studies. So obviously we're getting larger and larger data sets with hundreds or even thousands of subjects. Many of them are publicly available, so you'll soon be able to get the full 1,200 subjects from the human connectome, and there are thousands of subjects being collected for some of the follow-on human connectome studies. So as you may know, here at UCLA, we have a fairly large compute cluster sitting underneath our offices in brain mapping. And we have about 3,000 cores and about 408 nodes on those cores. And so we can use these fairly efficiently with our software, which we can run everything in about 3 hours on a typical data set. If you're running on the higher resolution HCP data, it takes much longer, but for more traditional acquisitions, it's fairly fast. And so we did an experiment a couple years ago where we just tried to see how fast we could do 500 scans. We were able to process them all in about 2.5 hours. And so not all of them worked, which is why we're interested in trying to identify these more rapidly. You can see a few cases when you look at the visualization. But in general, it worked very well, and you can do essentially a whole study, at least the computational part, in an afternoon. So what we're interested in working on, in addition to trying to improve the methods, are working on some tools to try and do quality control and try and help users identify cases where the software may have gone wrong so they can go back and make corrections. So I'm going to try and show the actual website if this works. So we've implemented this in JavaScript, and it's running on a web browser. And so you can actually see this is 100 different subjects, the data. So you can scroll through rapidly. You can look at different stages of the processing. And so we're trying to build this in a framework that we can distribute to individuals for use on their own systems. It's still in the works. Ultimately, we'd want to try and integrate it with some other systems, perhaps run it, for example, on Amazon Web Services. So you can easily find some cases where the software failed, and you can go back and look and see that the skull stripping was bad. And so that's caused a problem in the registration that then led to only finding a little blob of the brain instead of the whole thing. So if you went back and fixed the skull stripping, then hopefully this would fix itself. So that's one aspect of trying to improve the workflow. So we use this type of process a lot in our own experiments and our own studies. It's also a nice way of sharing data with collaborators as well, so you can have it on your website behind passwords if necessary, and send it to collaborators rather than sending them terabytes of results. So obviously we are interested in finding these errors automatically. So we do have in the plans to develop some interfaces that will try to look at the output of each stage of the processing and compare it with some statistical measures of how the software should have performed. So for example, if you're looking at the brain extraction, you might look at the total brain volume. Obviously, if it's only 100 cubic centimeters, then you probably did something wrong. If it's large enough to be the whole head, you probably did something wrong. Or it's possible that you have a really abnormal subject. In any case, you'd want to flag those results automatically and go back and review them. And we're also some of these ideas are going as well into the automated parameter selection that we're doing. So to automatically find the skull stripping parameters, for example, we're doing some iterative searches through the space based on a measure of surface area and brain volume. So these are the kind of things that we have in the pipe for the future. Another thing that we recently did, I don't know if a lot of you use NITP and Python. Yep. So we just, this past year I had a student working with me, Jason Wong, who developed the interface for it. And so far we've put the cortical surface extraction code into the main GitHub, the official NITP repository. So if you download NITP and you have BrainSuite installed, you can run the cortical surface extraction through NITP or any of the modules that are in it. We've also developed the interfaces for the diffusion programs and the registration programs, and we'll be testing them with the new release of the software and sending a pull request to the NITP developers probably within the week. And Jason also developed a set of workflows that you can actually run the whole BDP sequence and the SVReg sequence and the cortical extraction sequence in NITP in one session, and it's set up so it runs in parallel, so it will optimize the execution. And he's even gotten this to work on the Amazon Web Services, so if you wanted to run it on your local machine or on the cloud, it's possible. We don't have the instructions up on the site yet, but probably within, when he comes back from the summer break, because he's an undergrad. And he got a good internship, so. We'll be adding that to the main distribution so that you can run it on the cloud. One of the other things that we have for the NITP tools, he also developed a tutorial that's on our website. So it's in a Jupyter notebook, which basically it describes each step that he put together. He did a really nice job of documenting it. So it tells you how to run it. You can edit the code in the browser, and you can run it on your machine. I don't remember if I put the link in your slides or not, but so this is the notebook on the website, so you can look at it on the Jupyter page, and he described each step in the processing and how it works, and you can actually run this as well on your machine. So if you're interested in running BrainSuite in NITP, I'd recommend taking a look at it. It's still, this literally just went up on the site yesterday, so it is still in refinement, but if you're running issues with it, just let us know, because we're still actively developing it. So I'll just mention a few other areas where we're applying the software. Richard Leahy, who's my collaborator, has been very involved in MEG and EEG, and started the effort and continues to work on the effort to do source localization in a package called BrainStorm. And so we've developed tools, plugins for BrainStorm that allow you to import your data into BrainStorm so that you can take the cortical labelings that we have and put them into the software, and then use them to identify where a source activation may be in the code. So if you're doing MEG or EEG using BrainStorm, then you should check this out as well. And then another thing that we're looking at is and this is a collaboration with a group at the Cleveland Clinic, we're looking at taking the maps that we generate. So this is an example of taking the maps that we have and using them to map other data. In this case, we're taking the electrode positions from stereoelectroencephalography, where basically you have depth electrodes that are used for recording signals in patients with epilepsy. And we're using those to make an atlas of those signals and put them into a common space. And so the goal then is to try and use that information to try and get a better understanding of the process of epilepsy. And so you can see in the slide how the SIG positions are deformed by the registration. So this is an ongoing effort. I think they recently started the R01 for it. And so this is something that they're currently working on. And then I mentioned earlier that one of the reasons why we think our software tools, one of the strengths of our software tools is that we do have the capability to deal with brains that often have abnormalities or may have had a surgical resection, for example. So this is case, I believe, where the subject had part of the temporal lobe resected, and the segmentation software and the labeling software still managed to register and do a reasonable job of labeling it. And I have another example that So Young, who's joining us this afternoon to help with the lab, also did. So this is from some work she did a few years ago, where she was labeling a subject that had a very large lesion, and you can see the software was still able to do a reasonable job of labeling the areas around the lesion. And as I mentioned earlier, we do have tools that allow you to trace landmarks on the brain. So this is one possibility. If you have an area that's abnormal, you can trace landmarks on the atlas and the subject to try and constrain the registration in those areas. You could also, for example, trace landmarks in an area you're really interested in, such as the visual cortex, and then use that to get an improved registration. And then this is an example of a custom atlas. So I mentioned you can make your own atlases. So if you're interested in something other than human brains or other than adult brains, you can create a custom atlas. So we have some examples of pediatric lobe atlases that were developed by our collaborators. This is an example of a vervet atlas that was developed by Noor Al-Sharif, who worked in my lab about a year ago and is now a graduate student at McGill. And when she was working in the lab of Scott Fears, who's here at UCLA. And so she was helped us to define the process that is needed to produce an atlas from your own data and customize it. And so we refine those into a set of tools that if you do have your own data that you've labeled for an atlas, you can use that then in the registration process and label whatever data you're most interested in. So I'm going to jump out of MRI for a second and just mention some other areas that we're really interested in my group. So I'm sure all of you have heard of clarity imaging. If not, it's essentially a technique that allows you to turn brain tissue or other types of tissue optically clear, and you can have stains, either endogenous stains or immunohistochemistry stains that identify the neurons or other types of cells in the image. And so this is a section of a hippocampus from some data that was collected by Luis Del Torre, who's part of Dan Geschwin's lab here at UCLA. And you can see, we're looking at about less than a millimeter slice, and you can see volumetrically individual neurons, which I think is pretty amazing. And this is done with a confocal microscope. And we've they've also imaged some cortical neurons as well. So we're looking at these data sets and trying to apply some of the things we've learned in segmenting the MRI in the brain to try and segment these data as well. I'll show one more example. As that finishes spinning. And so this, I don't think this one is in your slide, but this is an example from Alan Mackenzie Graham, who's a professor in brain mapping as well. And this is the left cerebral hemisphere of a Thai-1 YFP mouse. And you can type in this URL if you feel like it, if you just want to see the data. So it's really pretty amazing, just what you can see. And this is a stack of about 100 slices that we did a maximum intensity projection through. So this is 1 millimeter of cortex. So just to give you some idea of the size of data that we're working at, we're trying to image around 1 micron resolution volumetrically. And so if you have a cubic centimeter of data, that's 1 terapixel. And so a mouse brain is probably maybe 1.5 cubic centimeters. So to do this kind of imaging, especially if you start acquiring 16-bit data or if you do multi-channel imaging, you're quickly pushing multiple terabytes just for one small piece. Of course, people would like to do this on whole human brains as well. And there's similar problems in some of the polarized light imaging methods where they're also acquiring just petabytes of data. So this is an interesting challenge for us as engineers and software developers to try and figure out how to process this amount of data and how to visualize it, as well as how to stitch it all together. And so these are some of the things that we're looking at now outside of MRI, but with the idea that we do want to try and bring this information back to MRI and make comparisons of what we see in the clarity data to what we see in the MR data. So that's pretty exciting. It's for me, we're still at a very early point in what we're doing, but we have done some work to try and cache images when we're in BrainSuite, so we can actually have data sets that are a few hundred gigabytes on disk that load fairly rapidly in the viewer. So that's something we're hoping to get out in the next year, probably as a separate tool from BrainSuite, but based on some of the similar concepts. And we are working as well on trying to do some automated segmentation of finding models of individual neurons, which is quite challenging because the signals here aren't nearly as clean as MRI, which makes it really difficult to find some of the objects, and particularly when they're crossing in various ways. So, but it's an exciting problem and one that we're really interested in. So I want to close there and just say that I hope that in the talk and in the lab that we'll be doing shortly, that our software tools provide a collection of flexible open source methods that you can use to analyze the brain imaging data. The highlights again are that we have fast tools for doing the image segmentation and surface extraction. We have unique tools for doing cortically constrained volumetric registration, as well as tools for doing all the diffusion registration, distortion correction, and tracking, and also tools for doing the group level analysis. Finally, I just mentioned again, we're actively continuing this project. We're developing new code all the time. And so if you do use it, or if you're interested in use it, we are really interested in hearing from you, and so you can contact us either at these addresses. These come to me as well as the rest of the team, or you can email me directly, of course, or you can post questions in our forums. So if you do use the software, I'd encourage you to be in touch with us. And finally, I'd just like to acknowledge again all the many collaborators on the project, as well as the people here at UCLA who contributed some of the data that you saw on the slides, and as well as the agencies that are funding the projects. And thank Mark and the rest of the community again for having me back, because it's been a great experience. So